Hello. This video is a preview of the lectures that you are going to see throughout the year as I record them live in my classroom. This is a mock lecture. I'm not actually speaking to any students online or anybody in person, but this is what they will look like, and it serves as a preview for what you can expect to see. In this video, what I'm going to do is my introductory lecture to biology. So we will be discussing the scientific method and science as it relates to biology, and also investigate a historical example of how an old scientific theory, spontaneous generation, was gradually overturned through better experiments that slowly revealed the truth. Let's get started. Okay, so the science of biology, part 1, first lecture for the year. So first of all, just talking about science in general and thinking like a scientist, I want you to think, well, what does science mean to you? What is the definition of that word, in your opinion? If you had to try to explain it to like a kindergartner. So explain it in very simple terms. What is science? Or maybe, why is science important to you? When we say, what does it mean to you? We could also mean that as well. And if you met a scientist, well, what do you think they would be like? Do you think they would be boring to talk to? Or do you think they would be exciting to talk to? Or do you think that all scientists are different? I mean, they're people, so you're gonna get different people. And then lastly, what do you think you have to do to be a scientist? What qualities does somebody who is a scientist need in order to be successful? We could also think, if you want to think like a scientist, which we are going to have to do in this class, and other people have to do that, a car mechanic, in order to figure out what's going on, has to think like a scientist. So the qualities and the behaviors and whatever attributions that scientists have, how do those apply and relate to other people and how we maybe should look at the world? So we'll begin with the scientific method. So the reason that science as a practice works is because it follows these two basic assumptions. The first is that the world is intelligible. Now, that may seem like a very obvious no-duh statement, but for a very long time, people assumed that certain things about the world, about the universe, just simply could not be known. For example, the reason that planets orbit. Nobody thought that you could understand that because the universe was supposedly created by someone that humans just couldn't possibly understand. And so nature would reflect that. So people didn't even bother. And people that tried, such as Galileo, well, they ended up being punished sometimes. And the other key thing is that the scientific process itself, it informs us of the truth. So truth doesn't come springing up from the ground, and it doesn't come falling out of the sky. And truth is not revealed in your dreams. Intuition and charismatic people and luck and things like that, those are not good. Those are not good methods of discovering the truth. The rigorous scientific process and the controlled experiment are the best ways we know of to uncovering what is true. So, the actual steps of the scientific method. The first step is always to ask a question, and it can be absolutely anything at all. Once you have a question in mind, once you have something in mind that you want to try to understand, you need to go out and make observations and gather information. And we call that information data. So data can be quantitative or qualitative. Quantitative data, that's the kind of data you can actually count. So if we want to count how many insects live in this particular square meter of ground, how many bird species live in this one tree, questions like that. Whereas qualitative data can really only be described. It can't necessarily be counted. So in nature, qualitative data might be something like we want to record animal behavior. So we want to look at maybe like the mating patterns of an animal, or how they react to predators or other animals. Something that you can't exactly put a number on. So once you have all of this data, and you should spend a very long time collecting data, then you can make inferences based on that data. So what an inference is that's a logical interpretation based on prior knowledge. So you use your observations and some, we could call it preliminary data, to make an inference, and you're going to make a prediction off of that. Because so far, we're just asking a question. We don't have a possible explanation for what's going on yet. But that is the next step. You generate your hypothesis. And a hypothesis is a possible explanation for a set of observations that is then validated or invalidated through experiment and observation. So if an idea cannot be validated, or in other words, if it just can't be proven or disproven, then it cannot be considered a scientific idea. And there are plenty of ideas that are not scientific. If something is subjective, then we really can't call it scientific. So, for example, who is the greatest guitar player who ever lived? A lot of people, probably most people that really know a lot about guitar, say it's Jimi Hendrix. But how do we actually, besides just asking people what their opinion is, how can we quantitatively decide that? We can decide it qualitatively, but it still really can't be considered a scientific idea. And if an idea or a statement is proposed that just simply cannot be overturned, such as a king saying, "My power, my authority is absolute and it is divine," or something like that, well, by the nature of that statement or argument, it can't be overturned. So it can't be considered a scientific idea, and it really shouldn't be regarded as a true idea either. It's what we call an argument from authority. And the real value of science is that scientific ideas can be proven wrong. Science is not dogmatic or authoritarian. Now, is the theory of gravity sort of, an established, definite truth? Well, yes, it is. But it was discovered, that truth was discovered by the scientific method, and it still can be proven wrong. Famously, Albert Einstein, he didn't prove Isaac Newton wrong, he just improved and revealed more information behind Isaac Newton's ideas, but they still reign true. Okay, the next step in our scientific method is going to be to test your hypothesis experimentally. If you look at old writings from like Aristotle or other ancient Greek philosophers, well, they came up with all kinds of ideas, but they didn't test their ideas. They just simply thought really hard about them. And if you read, they can say, well, we can obviously see why my opinion on this is right, because if you just think about it, blah, 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 blah. That's not scientific. Then once you have carried out your experiment, you need to analyze the data. Now, humans are actually very bad at reading data, and humans are just really bad at interpreting nature, sort of in general. But the math just simply can't lie. So here's an example of this. Suppose a certain disease afflicts 0.1% of the population. A test is devised to detect the disease. It has a false positive rate of 5%. So suppose a randomly selected person takes the test and tests positive. What is the probability that this person actually has the disease? Now, a lot of people would maybe guess like there's a 95% chance, because it says there's only a 5% error rate with the test. But it doesn't work that way. And if you don't have a mathematical model and a statistical model to dissect it, then it's almost impossible through just your logic, through what we think is common sense, to arrive at the correct answer. So this is a very hard problem, and don't feel bad about having no idea how to do it, because this question was famously posed to Harvard medical students, and 82% of them got it wrong. In fact, for the record, the correct answer is it's actually less than a 2% chance that the person actually has the disease. Now, I'm guessing you think that that answer is extremely counterintuitive, but that's kind of the point. A lot of information that we gain through science ends up being really counterintuitive because as human beings, we're flawed. We look at nature and we try to understand it, but even if we don't have a good way to really explain and test what we're seeing in nature, we just make up our own explanations. And that's a problem because it leads us to being wrong and holding on to ideas that are wrong for a long time. And I just don't think we can afford to do that forever. Now, the final step with all of this is to draw conclusions based on your data. So this is where you are going to revisit your hypothesis. Was I right or was I wrong? And in science, like in sports, like in learning in general, like in all kinds of things, you're going to be wrong many, many more times, or you're going to fail many, many more times than you are going to succeed. Even the best basketball players, the vast majority of them, they miss more shots than they make, for example. So science, another tremendous value in science is that the field is competitive. So when a discovery is made, other scientists actively try to disprove whatever one scientist discovered, or they try to improve on their idea. Now, this ensures that science is like a self-correcting enterprise, and scientists make their data public. They publish it. Universities and other organizations that do scientific research, that information is publicly available. For example, all of the discoveries that NASA has made, NASA is a government organization, so they make everything public. There are some classified things that NASA has because perhaps they're working side by side with the military on experimental propulsion or something like that. But the vast majority of all other discoveries, they're made publicly available. And now with mass printing and the internet, the access to information we have now is greater than it's ever been. And we see discoveries are being made and improved at a rate that is also greater than it has ever been. And these are very, very good things. Now, just to sort of prove that science has a very good track record, these are probably the two most important graphs in the world. The one on the left here is the percentage of the world population that lives in extreme poverty. 100 years ago, still 90% of all people in the world lived in extreme poverty. And today it's been completely flipped. Even though there are over 6 times as many people, close to 7 times as many people alive today as there were 100 years ago, 90% of them have escaped global extreme poverty. And this correlates very nicely side by side with the unprecedented raise in life expectancy as well. Humans live almost exactly, on average, twice as long as they did 100 years ago. You would maybe think that when you all of a sudden have 7 times as many people on your planet in a period of only 100 years, there won't be enough resources to go around, and people are going to starve, and people aren't going to be able to provide for themselves. But it's not just that the opposite of that has happened. The opposite plus a whole lot more has happened, and it's been very, very good for the human species. Another example, we could do this all day, but I'll just pick this one. Smallpox. So vaccines, how science improves the well-being of humans. It's not just discoveries to be like, oh, well, that's cool, but now what do I do? Science literally saves lives. So we can see, like in 1950, there were more than 500,000 cases of smallpox. But what you'll notice about this graph is that, and that, by the way, is fairly close to where we're at with COVID right now, but smallpox is far, far more deadly. Your odds of dying, no matter what age you are or underlying conditions from smallpox, was vastly greater than it is from COVID. But what you should notice about this graph is it only goes to 1977. That's because this virus, extremely deadly virus, which we're pretty sure killed somewhere around 300 million people from the year 1900 to 1977, this virus is extinct. Nobody on Earth has contracted it since 1977, and it's never coming back. Some other viruses like polio, we only see a handful of cases every year, but it still is there. But smallpox, maybe one of the absolute worst killers of the 20th century, science extinguished it. It does not exist at all. So throughout this class, we will be talking all the time about major discoveries and advancements and accomplishments that science has provided to give you an appreciation for it. There will be a lot of times this class will feel like a history class, and the reasons that science is important are historical, so they are worth talking about. But we will also be talking about how science is not always very readily accepted. So, for example, Charles Darwin in his time was very widely criticized. He's still criticized by quite an alarming number of Americans today. And there have been other instances where science was used not for progress, but for destruction. We could think of the nuclear bombings of Japan as one example. So whenever we can, we will bring up those examples in this class as well to talk about the role that science has in society, for better and for worse. Now, we'll move on and actually focus on biology here for the rest of this video. So, to set this up, for thousands of years, people thought that life, obviously living things are different, but people thought that life was powered by some invisible force, and that non-living matter could be turned into life. So we have the concept of like the vital essence, which is something that some Greek philosophers talked about. We have the concept of the soul, which thousands of religions all throughout the world came up with. We have the Force and the midichlorians from the Star Wars universe. What we will do today is we'll investigate how the scientific process gradually disproved this idea and proved that living things come from other living things. Now, real quick before we do that, let's talk about experimental design. So, as just a general example, let's say we want to test what is the best way to grow flowers, to get like the tallest flowers, for example. Well, we need to set up a test, an experiment to do that. So, plants need a lot of different resources. So that means there are many factors that affect the outcome of this experiment. And those are going to include the amount of sunlight, the color of the light. We could use artificial light, doesn't need to be sunlight. The amount of water that each plant is given, the type of fertilizer that they're given, the temperature that the plants are kept under, the pH of the soil, plus many, many, many other things. Now, if we want to, as a good scientific experiment, what we call a controlled scientific experiment is one where all of the variables, except for one, are kept the same. So for example, if we go back, we would only want to test one of these things. So maybe we want to know, well, which color of light produces the biggest, tallest flowers? Is it green light, red light, blue light, yellow light, white light, so on and so on? Well, that variable, the one that we change, is called the independent variable. It's purposely changed in the experiment. So we would have many, many, many different flowers, and we would grow each of them. All variables would be the same except for one. And then the dependent variable, well, that is the observed effect of the independent variable. So in this hypothetical experiment, the independent variable is the color of the light used to grow the plants, and the dependent variable is going to be the height of the plants, or maybe we could weigh the plants to track their growth. That is how a scientific experiment is set up. And one other thing that I don't have on here is you need to have a control group. You need to have a separate group in your experiment that is not subjected to any independent variable to make sure, because there can be errors in your experiment. So in this case, we would grow plants. You could do a control in a few different ways. You could use natural sunlight, because using colored light is the same as artificial. So we could test artificial versus natural light. Or a better control, and maybe we could have two controls, is to grow plants using absolutely no light whatsoever. So that would be an example of your control group, as it's called. And we actually do this kind of thing. So in the country of the Netherlands, they have, it's a tiny country, only the size of New Hampshire and Vermont put together. But they're the world's leading exporter in many types of foods, including tomatoes and chilies and several others. They grow their food scientifically indoors under controlled conditions, and they test and figure out what, if we change this variable or that variable, how does it increase or maximize our products here. And so they've been able to revolutionize the way they grow food this way. Okay. Also, when it comes to designing an experiment, there are some things which can't exactly be observed. So, for example, gravity. We can't actually see gravity, but we can observe the effects that gravity has. For example, if I hold this marker up and drop it, well, we just observed the effects of gravity. Amazing, right? Or one of the problems is we don't have the technology to observe certain things. Albert Einstein, more than 100 years ago, predicted, his math predicts that gravitational waves should be, they should exist from the collision of neutron stars or black holes. Well, it wasn't until 2017 that we had the technology to detect them, because the technology just had to be crazy sophisticated. And lo and behold, in 2017, we did find those. Or another problem is that people assume that some things can't be observed. And we sort of already talked about that example. So getting to specifically the biology here, everybody knew that you could find maggots on dead things. But weirdly, nobody knew where they came from, or nobody knew how they got there. Well, they had an idea. Their idea was that maggots would spontaneously appear on dead things. So, in other words, the dead animal, its tissue, its flesh, would turn into maggots. It's not that flies lay eggs, and that's where maggots come from. People thought it was this. And this is an idea called spontaneous generation. People thought that mice would just spring into existence out of bales of hay or piles of grain, as another example. People thought that worms just simply were made by the earth underground. It seems silly to us, but a long time ago, people didn't have any other explanation because they didn't go out there and actually observe nature carefully and test their claims. So here is a very, very basic experiment, one of the very first ones over the idea of spontaneous generation. And I think his first name was Francesco Redi. Here was his experiment. So he noticed that, well, you see flies on dead things before you see the maggots. So maybe that's where the maggots come from. So his hypothesis is flies produce maggots. So here's his very simple experiment. His control group is the uncovered jars, and then his experimental group, or whatever, his group with the independent variable is the jars that are covered up. So flies cannot get into there. And lo and behold, not very surprising to us, and here they call the independent variable the manipulated variable. means the same thing. Well, lo and behold, there were no maggots on the covered up jar. So to Reddy, this disproved the idea that meat produces maggots. Cool. So this is an example of how a previous idea gets overturned. However, we're nowhere close to done with this idea yet. So a very important thing, the way science works is that everybody is trying to prove everybody wrong. And so another scientist comes along named John Needham, and he tries to disprove Reddy. So his goal was to revalidate spontaneous generation theory. So what Needham did was, he didn't use meat, but he used boiled flasks containing broth so that it would sterilize them, killing any potential life that was inside of those flasks. And then he just left them out. And after a few days, the flasks were swarming with microorganisms. So according to Needham, because he didn't have the technology to see otherwise, according to Needham, the broth turned into microorganisms. Then we have Lazaro Spelanzani, fantastic name. He tries to modify Needham's experiment to disprove him. Again, this is how science works, and it's one of the great strengths of it. So Spelanzani modified the experiment, and he made air the independent variable. So two flasks, they're both boiled, but then one is left open, just as it was in Needham's experiment, and the other, he heats up the glass and folds it and pinches it shut. And as we can plainly see in that glass, no life ever develops. Now, Spelanzani did not, despite the results of this experiment, Spelanzani still thought that spontaneous generation happened. He just thought that air was the thing that produced it. So the air literally breathes life into the gravy. Now, that's sort of true, because the microorganisms, the germs or whatever, they get there from the air, but they already exist in the air. They float into there. It's not that the molecules of air turn into life. In the air, there is life. It's just too small for us to see. Then finally, the last iteration of this experiment, Louis Pasteur, who is certainly one of the most important people who ever lived. He came up with techniques that sterilize and have saved countless lives. We call it pasteurization because of him. He figured out a new way to design the experiment to still test the same claim, but to make sure that the experiment is as valid as possible. So this left no reasonable doubt as to whether or not spontaneous generation is possible. So in his experiment, he has this sort of curved neck on the flask, so air can get in there, but any microbes floating around in the air, they're going to get stuck and not be able to take hold inside of that broth. And so these flasks were left alone for periods of at least a year or more, and they were still sterile on the inside. Then you break off that stem so that there's a very easy access to the broth on the inside, and very shortly, it's swarming with microorganisms. So this proved that the life is already there. It exists. Maybe we can't see it, but it's definitely there. Mold is like this. You don't see it, and then all of a sudden, you look in the fridge and something's just covered in the stuff. It's always there. Spontaneous generation just isn't true. Now, this is why experiments have to keep being performed, because one person comes up with a new way to test a claim, and then we can finally get some insight here. The Louis Pasteur experiment, that's been done many different ways, many different times. And I should probably point out, I wrote it that way intentionally. Left no reasonable doubt. Now, spontaneous generation was effectively disproven. Now, do we have to test the claim against spontaneous generation forever, countless times, under every single example we can think of forever to draw a conclusion? No. Now, outside of the world of mathematics, can we know anything absolutely for sure? Well, no, probably not. But we don't need to, because if we had that attitude, then we could just run around saying nothing is true, everything is permitted, which I think that's the slogan to the Assassin's Guild in Assassin's Creed. But our heads would explode if we lived in that world. So we get to the point of leaving no reasonable doubt. So experiments like Pasteur's have been carried out, and they've basically been confirmed. So what happens when similar experiments always produce the same results? Well, the underlying hypothesis, it graduates and becomes a scientific theory. So theories in science, these are regarded as facts, but they are subject to change because they are tested and confirmed experimentally. And if new and better and different experiments show different results, then we will modify our theory. But some theories, that just isn't going to happen. So the theory of gravity, that's just plainly a fact. The germ theory of disease. We can't really watch a bacteria give you pneumonia, but we know that that's what bacteria do. The theory of relativity, again, that's something that you can't directly see it, but you can observe the effects of it. The theory of evolution by natural selection, that is a fact, despite what quite a lot of people in America think. That's another thing that you can't exactly watch it happen right before your eyes, but we do have gobs and gobs of evidence for it. And atomic theory. Atoms cannot be seen, and it's not that we don't have the technology to see atoms, it's that the technology will very likely never exist because atoms are smaller than any wavelength of energy that could possibly interact with them directly. We can see the outline of atoms using really sophisticated, what are called scanning electron microscopes, but that's as good as it gets. But through different experiments, we can still know that atoms do exist. And that really is the tremendous value of science. It teaches us so much about the world, and it reveals to us things that were there all along, but we would have never known existed if we didn't approach them in a scientific way. Thank you for watching.